We're coming now to the end game of replication. For circular DNAs, in bacteria specifically, the leading and lagging strands are completed when all the RNA is removed and ligase seals the ends. It's circular, so once around the circle, and it's complete. All of the sequences in bacterial DNA are replicated without any loss of information because these DNAs are circular. On the other hand, eukaryotic DNAs are linear, and they have something called telomeres. So what happens at the ends of these linear DNAs? If you think it through and draw it out, you cannot make an RNA primer at the 5' end. Remember, you're copying 5' to 3', so you have to lay down a 5' to 3' RNA primer, and that has to be removed. So there is always going to be some unreplicated section at the end of linear DNA. So the DNA strands would get shorter and shorter with each replication. Eukaryotes manage this very difficult and specific problem using a special structure, and these are termed telomeres. And there's an enzyme, telomerase, that utilizes these telomeres, and it contains an internal RNA component that is able to prime replication at the 5' end and extend it. Without telomerase, the telomeres at chromosome ends would shorten. Telomerase extends the 3' end using an RNA template that is part of the protein itself. The 5' end of the complementary strand may be digested to ensure that the 3' end is accessible to move into the telomerase active site to extend the DNA and prevent loss. The RNA template within the enzyme provides a mechanism for adding DNA sequence to the 3' end, which is followed by priming and synthesis to produce double-stranded DNA. Looking at this process, you have the parental strand with an incomplete, newly synthesized lagging strand. The 5' most RNA primer and DNA extension. But you simply can't get to the end because the RNA is taken away, leaving a segment of DNA that cannot be copied. The solution is to expand 5' to 3', copying the bound RNA template. The telomerase slides along and copies this sequence multiple times to extend the 3' end of the DNA sufficiently that the complement can be primed and synthesized and the original DNA is preserved. And sometimes you actually get extension of the DNA sequence. So the telomere may elongate or shorten depending on the activity of telomerase. This is a very potent way of preserving DNA. Telomerase levels are regulated by the cell and influenced by environmental and cellular factors. In most cells, the telomeres slowly become shorter, as if there is a kind of planned obsolescence. This effectively produces a count of the number of cell divisions. Often, if you look at cells in an organism over time, you can see the loss of telomeres as part of the aging process. Looking at mice has shown us that this shortening of telomeres actually has a role in regulating cell division. In fact, this planned obsolescence doesn't apply in all cells. It's not true, or mostly not true, in bone marrow, skin, stem cells, and especially not true in cancers. In these cells, often telomerase levels are enhanced. The rapid and continued cell division in cancers would result in the telomeres getting shorter. But these cells often have just enough telomerase activity to protect the DNA ends. Knowing how much is right is not clear, but current research efforts are investigating chemotherapies to target telomerase. And telomerase is a major target for life extension supplements.